Welcome to the next session of the lecture. We are considering now which is a stronger variant of the lac promoter. This continuation is of the table of the milestone, the TAC promoter, a functional hybrid derived from the TRP and lac promoters, right? And now Debo and his colleagues also knew that the lac UB5 promoter, which is a strong variant of the lac promoter, had a consensus sequence at its 10 but not its 35 region, while the TRP promoter, which normally controls the transcription of genes involved in the biosynthesis of tryptophan, has a consensus sequence at its 35 but not at its 10 region. They decided to create a fusion promoter which included the 10 region from the lac promoter and the 35 region from the TRP promoter. They tested this new TAC promoter, as they call it, for its ability to direct the synthesis the enzyme galactose kinase in E. coli and compared it in the same assay system with the TRP and lac promoters. In agreement with their initial idea, the TAC promoter was found to be approximately 5 times stronger than the TRP promoter and 10 times stronger than the lac promoter. In addition, like the lac promoter, the lac promoter was repressed by the lac repressor and de-repressed by the IPTG. Thus, this new promoter was not only strong but also reglutable. Thus, for each clone gene, it is important to establish that the ribosome binding site is properly placed and that the secondary structure of the mRNA does not prevent its access to the ribosome. A number of convenient vector systems that incorporate both transcription and translation signals for the expression of cloned eukaryotic genes in E. coli have been developed. One such system is called expression vector PKK2332 and includes a number of elements including the following in figure 6.19, an ampicillin-resistant gene as a selectable marker, the TAC promoter, the LAGZ ribosome binding site, an ATG start codon located 8 nucleotides downstream from the ribosome binding site, and the transcription terminators T1 and T2 from the bacteriophage lambda. And the clone gene is inserted into an NCOL or NCO1, PST1 or HIND3 site that lies between the ribosome binding site and the transcription terminator so that it is in the same reading frame as the ATG start codon. After induction and transcription, the mRNA of a clone gene is efficiently translated. However, since the nucleotide sequence that encode the amino acid in the N-terminal region of the target protein vary from one gene to another, it is not possible to design a vector that will eliminate the possibility of mRNA foldback in all instances. Therefore, no single optimized translational initiation region can guarantee a high rate of translation initiation for all clone genes. Consequently, the expression vectors described above are merely starting points for the optimization of translation initiation. A cellular incompatibility that can interfere with efficient translation occurs when a clone gene has codons that are rarely used by the host cell. For example, ACGGG and the rest are shown here are the least used codons in E. coli. In these cases, the host cell may not produce enough of the transfer RNAs, tRNAs that recognize these rarely used codons, and the yield of the clone gene protein is much lower than expected. An insufficient supply of certain tRNAs as may lead to either a reduction in the amount of protein synthesized or the incorporation of incorrect amino acids into the protein. Any codon that is used less than 5 to 10% of the time by the host organisms may cause problems, particularly detrimental to high levels of expression, or places where 2 or more rarely used codons are close or adjacent or appear in the N-terminal portion of the protein. There are several experimental approaches that can be used to alleviate this problem. 1. If the target of a gene is eukaryotic, it may be cloned and expressed in a eukaryotic host cell. 2. A new version of the target gene containing codons that are more commonly used by the host cell may be chemically synthesized, codon optimization. 3. A host cell that has been engineered to overexpress several rare tRNAs may be employed, as shown in figure 6.20. In fact, an E. coli strain that overproduces the tRNAs ARG-U, ILY, and LU-W, which are specific for the codons, AGG, AGA, and the rest, respectively, is available commercially. This cell line is sold for the explicit purpose of expressing a high level of foreign protein that use these rare E. coli codons. With this commercially available E. coli cell line, it is possible to overexpress the ARA-H2 protein, a peanut allergen approximately 100-fold over the amount that was synthesized in conventional E. coli cells. Using the approach described, it should be possible to produce large quantities of a variety of heterologous proteins that are otherwise difficult to express in E. coli hosts. Figure 6.20 is a schematic representation of the expression of foreign proteins in a typical E. coli host cell A, and in an E. coli host cell that has been engineered to overexpress several rate tRNAs B. Have a look at the representation. Take a screenshot and make your notes right. Increasing protein stability. In recent years, it has become almost routine to overproduce a wide range of different foreign proteins in E. coli or other host organisms. However, to recover large amounts of purified active protein requires that the protein be as stable as possible. In this regard, there are certainly large differences in the intrinsic stabilities of different proteins. In addition, a protein produced in a heterologous host cell may, for various reasons, be less stable than the same protein produced in its normal cellular environment. Intrinsic protein stability. Intrinsic protein stability. Under normal growing conditions, the half-lives of different proteins range from a few minutes to hours. The basis for this differential stability is both the extent of disulfide bond formation and the presence of certain amino acids at the N-terminus. For example, when specific amino acids were attached to the N-terminus of beta-galactosidase, the in vitro survival time of the protein varied from approximately 2 minutes to more than 20 hours, as shown 6.4. Table 6.4. Amino acid additions that extend the intrinsic survival of proteins can be readily incorporated into clone genes. Often the presence of a single extra amino acid at the N-terminal is sufficient. To stabilize the target protein, probably by making it less susceptible to degradation by certain cellular proteases, and long-lived proteins can accumulate in cells and thereby increase the yield of the product. This phenomenon occurs in both prokaryotes and eukaryotes. Going back to the table, let's see the details. Table 6.3 shows increases in gene expression and that result from altering the codon usage of the wild-type gene for cDNA to more closely correspond to the host E. coli cell. So the protein and the improvement fold is shown here. So the proteins are human interleukin-2, glycerate interferonide tetanus toxin fragment C, human cardiac troponin T, mouse C force protein, spinach plastocyanin, human neurofibromin, human glutathione transferase M22, human phosphatidylcholine transferase protein, human interleukin-6, human interleukin-18, and plasmodium vaccine candidate antigen. In some cases, only a small number of codons were altered, while in others, the entire gene was synthesized with the optimal codon usage for expression in E. coli. Now going over to the subject matter proper again, in contrast to the amino acids at the N-terminus, which can increase the stability of a protein, there are internal amino acids sequences that make a protein more susceptible to proteolytic degradation. These regions of the protein, which are called PE or best sequences, are rich in proline, glutamic acid, serine, and threonine. They are often but not always flanked by clusters of positively charged amino acids such as lysine and arginine, and may act to mark proteins for degradation within the cell. In some instances, it is possible to enhance the stability of protein by altering its best regions by genetic manipulation. Such changes, of course, must not alter the function of this target protein. Table 6.4 shows stability of a beta-galactosidase with certain amino acids added to its N-terminus. Amino acid added and the half-life is shown. So these are details you can just take a screenshot and you should be able to make notes. Now this is figure 6.21, which shows a regulation of the synthesis of thioreduction target protein fusion in the absence A or presence B of tryptophan in the growth medium. The arrows label P, TRP, and PL indicate the direction of transcription. OTRP, the operator region where the TRP repressor protein binds. O1, the operator region where the cell repressor binds. PTRP, the TRP promoter. PL, the leftward promoter from bacteriophage lambda. TT transcription termination region. The box between the thioreduction and the target genes indicates the tRNA region that codes for the peptide that acts as the enterokinase cleavage site. The horseshoes indicate the binding of a repressor protein to its operator region. This is a picture of the representation as read up just now. Right? Now, facilitating protein folding. Many of the proteins that are produced in E. coli accumulate in the form of insoluble intracellular biologically inactive inclusion bodies. Although a small amount of biologically active protein can often be recovered from inclusion bodies, the extraction procedure requires expensive and time-consuming protein solubilization and refolding procedures. Since the insolubility of proteins is often a consequence of their folding, various strategies have been devised to avoid this problem. For example, fusion proteins that contain thioreduction, an 11.7 kilodalton protein, as the fusion partner that contain remain soluble even when up to 40% of the cellular protein consists of the fusion protein. When this system, the target gene is cloned into a multiple cloning site just downstream from the thyroreduction gene. Both of these genes are under the control of the P1 promoter on an E. coli plasmid vector. The E. coli host shell for this system has a CL repressor protein construct that consists of a copy of the gene for the CL repressor protein under the transcription control of the TRP promoter integrated into the chromosomal DNA. In the absence of tryptophan, as shown in figure 6.21a, a sufficient amount of repressor gene, CL repressor protein, is synthesized to prevent transcription from the P1 promoter and therefore prevent the production of the fusion protein. When tryptophan is added to the growth medium, figure 6.21b as shown, the TRP promoter is turned off by the TR repressor. The CL repressor protein is not synthesized, and the DNA construct encoding the fusion protein is transferred from the P1 promoter, the plasmid, and then translated. In this case, the fusion protein, which consists of the thioreduction and the target protein, is synthesized and accumulates preferentially at osmotically sensitive sites called adhesion zones at the inner periphery of the host E. coli cytoplasmic membranes. The soluble fusion protein is selectively released by osmotic shock from E. coli into the growth medium. If required, the native form of the target protein can be released from the fusion protein by treatment with enzyme enterokinase. Finally, since thyroreduction is stable at temperatures as high as 80 degrees centigrade, in those instances where the target protein is also stable at higher temperatures, the fusion protein may be purified by high temperature incubation, a condition that denatures most of the other cellular proteins. In E. coli and other gram-negative bacteria, the disulfide bond formation takes place in the periplasmic space, which is topologically equivalent to the endoplasmic reticulum in eukaryotes, but is a much more oxidizing environment. Disulfide bond formation in E. coli requires the participation of two soluble periplasmic enzymes, DSBA and DSBC, and two membrane-bound enzymes, DSBB and DSBD. Unfortunately, foreign proteins that contain 3 or more disulfide generally do not fold correctly in bacteria and often form inclusion bodies. In these instances, the foreign protein is usually produced in animal or plant cells. However, researchers have devised some strategies to overcome this problem of recalcitrant proteins with a large number of disulfide bonds. Human tissue plasminogen activator is an important therapeutic protein that is used to remove blood clots. The active form of this protein is a 527 amino acid serine protease that folds into 5 distinct structural domains with 35 cysteine residues that participate in the formation of 17 disulfide bonds. The cDNA for human tissue plasminogen activator was cloned downstream of a DNA sequence that encodes a leader peptide. It was previously used to facilitate the secretion to the periplasm of other eukaryotic proteins. However, in this case, only trace amounts of human tissue plasminogen activator were produced. This low level of activity is thought to reflect the fact that this complex protein is unable to fold properly in E. coli. In an attempt to increase the yield of human tissue plasminogen activator, the gene was co-expressed with the gene for either a rat or a yeast protein disulfide isomerase, both of which has been found to assist protein folding in other cases. However, this strategy did not affect the amount of active human tissue plasminogen activator that could be obtained. On the other hand, the co-expression of high levels of DSPC, figure shown as 6.22, resulted in more than 100-fold increase in the production of functional human tissue plasminogen activator. Alterations in the level of the other DSP proteins did not affect the amount of active human tissue plasminogen activator that could be recovered. To realize the maximum benefit from DSPC co-production, it was necessary to induce the synthesis of this protein approximately 30 minutes prior to the induction of human tissue plasminogen activator. Similarly, research in another area found that while DSPC overproduction had the largest impact on promoting the correct folding of horseradish peroxidase in E. coli and preventing its aggregation, the simultaneous overproduction of all 4 DSP proteins yielded the greatest amount of properly folded and active horseradish peroxidase. Thus, the experience of several researchers clearly demonstrates that for correct folding and disulfide bond formation of eukaryotic proteins produced in E. coli require high levels of the DSP proteins and possibly other protein disulfide isomerases. Co-expression strategies. Co-expression strategies. The expression of some thermolipile foreign proteins in E. coli host strains, which are typically grown at 37 degrees centigrade, often results in the formation of inclusion bodies of inactive protein. This occurs because the foreign protein misfolds when it cannot attain its native active conformation. A variety of strategies have been developed, albeit with limited success, to circumvent this problem. Cultivation of recombinant strains at low temperatures, which is beneficial to proper protein folding, often significantly increases the amount of recoverable active protein. However, mesophilic bacteria like E. coli grow extremely slowly at low temperatures. In one study, the chaperonin 60 gene CPM60 and the cocperonin 10 gene CPM10 from the psychrophilic bacterium Polyspira antarctica were introduced into a host strain of E. coli, with the result that the E. coli strain gained the ability to grow at a high rate at low temperatures, 4 to 10 degrees centigrade. This strain was subsequently transformed with a plasmid encoding a temperature-sensitive esterase. The expression of temperature-sensitive esterase in the E. coli strain carried the 2 chaperone genes at 4 to 10 degrees centigrade yielded esterase specific activity that was 180-fold higher than the activity from the native E. coli strain without chaperonins grown at 37 degrees centigrade. Interestingly, and contrary to expectation, the psychrophile chaperonins do not facilitate the proper folding of the esterase and do not affect its catalytic properties. Although very high levels of expression of the cloned esterase were not attained, this work is an important first step in the development of expression systems for proteins that are sensitive to high temperature and might otherwise be difficult to produce. The next logical step in the development of the system would likely be the construction of an E. coli host cell that contains stably integrated copies of these chaperone genes in the chromosome. The E. coli and most of the microorganisms that are used to express foreign proteins generally require oxygen for optimal growth. Unfortunately, oxygen has only limited solubility in aqueous media. As all the cell density of a growing culture increases, the cells rapidly deplete the growth medium of dissolved oxygen. When cells become oxygen limited, exponential growth slows, and the culture rapidly enters a stationary phase, during which the cellular metabolism changes happen. One consequence of the stationary phase is the production by the host cells of proteases that can degrade foreign proteins. Oxygen dissolves into the growth medium very slowly, so this problem is not always alleviated when large amounts of air or oxygen are added to the growth medium, even with high strain rates. Modification of the fermenter configuration to optimize the aeration and agitation of cells and addition of chemicals to the growth medium to increase the solubility of oxygen have been tried in an effort to deal with the limited amount of dissolved oxygen. However, these efforts have met with only limited success. Use of protease deficient host strains. Protease deficient host strains. Now, one possible way to stabilize foreign proteins produced in E. coli is to develop host strains that are deficient in the production of proteolytic enzymes. However, this is not as simple as it might appear. E. coli has at least 25 different proteases, and only a few of them have been studied at the genetic level. Moreover, these proteases are important for the degradation of abnormal or defective proteins, which is a housekeeping function that is necessary for the continued viability of the cells. When one studies strains with mutations in one or more protease genes were constructed, generally the strains that were most deficient in overall protease activity grew most slowly, thus decreasing protease activity caused cells to be debilitated. However, an E. coli strain with mutations in both the gene for the RNA polymerase sigma factor that is responsible for heat shock protein synthesis, RBOH, heat shock protein synthesis. And the gene for a protease that is required for cell growth at high temperatures, DEGP, secreted target proteins that had a 36-fold greater specific activity that when they were produced in wild-type host cells. This increase in activity reflected decrease in the proteolytic degradation of these secreted proteins. Bacterial hemoglobin. Some strains of the bacterium, a gram-negative obligate aerole, normally live in oxygen-poor environments, such as stagnant ponds. To obtain a sufficient amount of oxygen for their growth and metabolism, these organisms synthesize a hemoglobin-like molecule that binds oxygen from the environment and increases the level of available oxygen inside cells. When the gene for this protein was cloned and expressed in E. coli, the transforms displayed higher levels of synthesis of both cellular and recombinant proteins, higher levels of cellular respiration, a higher ATP production rate, and higher ATP contents, especially at low levels of oxygen in the growth medium, than the non-transformed cells. In these transformants, the vitreocellular hemoglobin increases the intracellular effective oxygen concentration, which raises the activities of both cytochrome D and cytochrome O. This causes an increase in proton pumping, with the subsequent generation of ATP that were providing additional energy for cellular metabolic processes. For this strategy to be effective in different host cells, not only must the reduced hemoglobin gene be efficiently expressed, but the host cells must also be able to synthesize the heme portion of the hemoglobin molecule. Once these conditions have been met, this strategy can be used to improve growth as well as foreign gene expression in a range of different industrially important bacteria, including E. coli, Streptomyces spp, Enterobacter aerogenes, and Xanthomonas maltophilia. Next we have is limiting biofilm formation. So, limiting biofilm formation. Okay. Right, now in many natural environments, bacteria are commonly found to be associated with solid surfaces and only rarely exist as free-swimming entities. The bacterial cells typically attach to a surface, form a monolayer, and later organize into a biofilm. A mixture of bacterial cells and polysaccharides, typically alginate, that may be as much as 200 to 200 micrometers thick. When they are part of a biofilm, bacterial cells are generally protected against hostile agents in this environment, such as biocides, bacteriophages, and protozoa. Bacterial cells that form biofilms or otherwise produce significant amounts of extracellular polysaccharide are difficult to transform with plasmid DNA and are typically resistant to high levels of antibiotics, which find it difficult to enter the bacterial cells. These cells are limited in the amount of foreign proteins that they can produce, probably reflecting the large amount of cellular resources directed toward polysaccharide synthesis and the limited amounts of nutrients that are able to enter cells within the biofilm. Now, figure 6.23 is a schematic representation of the binding of O2 by vitreocellular hemoglobin. The utilization of this O2 in pumping by proteins such as cytochromes H+ from the cytoplasm to the periplasm, and the subsequent coupling of H+ uptake by ADPase to ATP generation, ADP, adenosine diphosphate. The figure shows that. Please take a screenshot, prepare your notes. The table here shows the use of vitreocellular hemoglobin for improved production of foreign proteins in bacteria. Right, now we have the type of improvement: increased protein production, then the growth in alpha amylase production with E. coli bacterium, total protein content, protein secretion, alpha amylase production, increased chemical production, increased antibiotic production, where you have cephalosporin C, actin Browden, erythromycin, growth and remnant production, monensin and chlor-tetracycline, and hence bioremediation, growth and degradation 2,4-dinitrotoluene, degradation of benzoic acid, growth and degradation of benzoic acid, degradation 2-chlorobenzoate, and hence philosophy adapted from Zhang et al. Credits to them. Use of protein deficient host strains. One possible way to stabilize foreign proteins produced in E. coli is to develop host strains that are deficient in the production of proteolytic enzymes. However, this is not as simple as it might appear. E. coli has at least 25 different proteases. Right, and only a few of them have been studied at the genetic level. Moreover, these proteases are important for the degradation of abnormal or defective proteins, which is a housekeeping function that is necessary for the continued viability of the cells. And one study strains with mutations in one or more proteases genes were constructed. Generally, the strains that were most often deficient in overall protease activity grew most slowly, thus decreasing protease activity, causing cells to be debilitated. However, an E. coli strain with mutations in both the gene for the RNA polymerase sigma factor that is responsible for heat shock protein synthesis, RPOH, and the gene for a protease that is required for cell growth at high temperatures. DGP secreted target proteins that had a 36-fold greater specific activity than when they were produced in wild-type host cells. This increase in activity reflects a decrease in the proteolytic degradation of these secreted proteins. Bacterial hemoglobin. Some strains of the Vetuicella bacterium, a gram-negative obligate aerobe, normally live in oxygen-poor environments, such as stagnant ponds, to obtain a sufficient amount of oxygen for their growth and metabolism. These organisms synthesize a hemoglobin-like molecule that binds oxygen from the environment and increases the level of available oxygen inside cells. When the gene for this protein was cloned and expressed in E. coli, the transformants displayed higher levels of synthesis of both cellular and recombinant proteins, higher levels of cellular respiration, a higher ATP production rate, and higher ATP contents, especially at low levels of oxygen in the growth medium, than did non-transformed cells. In these transformants, the vitreocellular hemoglobin increases the intracellular effective oxygen concentration, which raises the activities of both cytochrome D and cytochrome D-O. This causes an increase in the proton pumping, generation of ATP, thereby providing additional energy for cellular metabolic processes. For this strategy to be effective in different host cells, not only must the vitreocellular SP hemoglobin gene be efficiently expressed, but the host cell must also be able to synthesize the heme portion of the hemoglobin molecule. Once these conditions have been met, this strategy can be used to improve growth, as well as foreign gene expression in a range of different industrially important bacteria, including E. coli, Streptomyces spp, Enterobacter aerogens, and Xanthomonas maltophilia. Limiting biofilm formation. In many natural environments, bacteria are commonly found to be associated with solid surfaces and only rarely exist as free-swimming entities. The bacterial cells typically attach to a surface, form a monolayer, and later organize into a biofilm. A mixture of bacterial cells and polysaccharides, typically alginate, that may be as much as 100 to 200 micrometer thick. Figure 6.24 shows that. When they are part of a biofilm, these bacterial cells are generally protected against hostile agents in the environment, such as biocides, bacteriophages, and protozoa. Bacterial cells that form biofilms or otherwise produce significant amounts of extracellular polysaccharide are difficult to transform with plasmid DNA and are typically resistant to high levels of antibiotics, which find it difficult to enter the bacterial cells. These cells are limited in the amount of foreign protein that they can produce, probably reflecting the large amount of cellular resources directed toward polysaccharide synthesis and the limited amounts of nutrients that are built to enter cells within the biofilm. This figure 6.23 shows schematic representation of the binding of O2 by vitreocellular hemoglobin, the utilization of this O2 in pumping bioproteins such as cytochromes, H plus from the cytoplasm to the periplasm, and the subsequent coupling of H plus uptake by ATPase to ATP generation, ADP, and adenosine diphosphate. The figure shows that, right? Now, workers have previously tried to limit the formation of biofilms either by adding biofilm inhibiting chemical agents to the bacterial growth medium or by utilizing new materials to which bacteria adhere less efficiently. However, these approaches have met with only limited success. Another way to limit biofilm formation is to genetically engineer bacterial strains that are deficient in biofilm formation. This was done by creating an E. coli strain in which the genes that were involved in the biosynthesis of cholinic acid, and the bacterial pili were deleted. Pili are required for initial attachment of a bacterial cell to a solid surface. Coli are needed for cell-cell and cell surface attachment, and cholinic acid contributes to the three-dimensional structure of the biofilm. When genes involved in these functions were deleted, the resultant strain of E. coli was unable to form biofilms, displayed an increased sensitivity to antibiotics, transformed with a much greater efficiency, and was able to produce a higher level of recombinant protein, as shown in 6.6. This result indicates that it is possible to engineer an E. coli host cell to be more efficient at producing foreign proteins. In addition to producing higher levels of recombinant proteins, the use of a bacterial host strain that displays substantially increased sensitivity to common antibiotics may obviate the overuse of antibiotics in the production of foreign proteins. This should make it easier to ensure that the final purified protein products is free of any contaminating antibiotics. The next is DNA integration to host chromosome. A plasmid imposes a metabolic load on the cell because of the energy that is used for its replication and for the transcription of RNA and translation. Table 6.6 shows the performance and behavior of wild type compared to biofilm minus E. coli. It shows the strain, the percentage adherence to polystyrene surface, sensitivity to streptomycin, sensitivity to rifampin, transformation efficiency, number of transformation per microgram of plasmid DNA, intercellular endoxylinase activity, extracellular lipase activity. And of the proteins that it encodes, for the most part, high copy number plasmids impose a greater metabolic load than to low copy number plasmids. As a consequence, a fraction of the cell population often loses its plasmids during cell growth. Also, cells that lack plasmids generally grow faster than those that retain them, so plasmidless cells eventually dominate the culture. After a number of generations of cell growth, the loss of plasmids containing cells diminishes the yield of the cloned gene product. Investigators have devised at least two methods of combating this loss. On a laboratory scale, plasmid-containing cells are maintained by growing the cells in the presence of either antibiotic or an essential metabolite that enables only plasmid-bearing cells to thrive. But the addition of either antibiotics or metabolites to pilot plant or industrial scale fermentation can be extremely costly, and it is imperative that anything that is added to the fermentation, such as an antibiotic or a metabolite, be completely removed before the product is certified as is fit for human use. Moreover, for genetically engineered microorganisms that are designated to be released into the environment to remain both effective and environmentally safe, it is essential that the cloned DNA be retained and be neither easily lost nor transferred to other microorganisms. For these reasons, the introduction of cloned DNA directly into the chromosomal DNA of the host organism can overcome the problem of plasmid loss. When DNA is part of the host chromosomal DNA, it is relatively stable and consequently can be maintained for many generations in the absence of selective agents. The chromosomal integration site of a cloned DNA must not be within an essential coding gene. Consequently, the input DNA sequence must be targeted to a specific non-essential site. Within the chromosome, in addition, targeted to a specific non-essential site within the chromosome. In addition to ensure efficient production of the target protein, the input gene should be under the control of a regulatable promoter. For the integration of DNA into a chromosomal site, the input DNA must share some sequence similarity, usually at least 50 nucleotides, with the chromosomal DNA, and there must be a physical exchange recombination between the two DNA molecules. Briefly, a generalized protocol for DNA integration includes the following steps. Identify the desired chromosomal integration site, that is a segment of DNA on the host chromosome that can be disrupted without affecting the normal functions of the cell. Isolate and clone part or all of the chromosomal integration site. Ligate a clone gene and regulatable promoter either into, as shown in figure 6.25a, or adjacent to figure 6.25b, the cloned chromosomal integration site. And for transfer the chromosomal integration, fragment clone gene construct into the host cell as part of a plasmid that cannot replicate in the host cell. Select and perpetuate host cells that express the clone gene. Propagation of the clone gene can occur only if it has been integrated into the chromosomal DNA of the host cell. When a host cell is transformed with a non-replicating plasmid that carries the clone gene in the middle of a portion of the cloned chromosomal integration site, the DNA and the plasmid can base pair with identical sequences on the host chromosome, as shown in figure 6.25a. The integration occurs as a result of host enzyme catalyzed double crossover, alternatively a single crossover that incorporates the entire input plasmid into the host chromosome may occur, but not shown here. Similarly, insertion of the entire input plasma DNA occurs when the clone gene is inserted next to the cloned chromosomal integration site, by shown in 6.25b. The effectiveness of integration of a clone gene was examined in B. subtilis. The investigators constructed an E. coli plasmid that contained an alpha amylase gene from Bacillus amino liquefaciens that had been inserted into the middle of a chromosomal DNA fragment from B. subtilis but could not replicate in B. subtilis. This construct, however, could transform B. subtilis transformants expressing alpha amylase, an enzyme-derived alpha amylase gene had been enzyme involved in the hydrolysis of starch were recovered, indicating that the alpha amylase gene had been integrated into the B. subtilis chromosome and was functioning. The selected transformants were resistant to ampicillin and chloramphenicol because both of the antibiotic-resistant genes were on the input plasmid. This result indicated that a single recombination event must have occurred and caused the integration of the entire plasmid into B. subtilis chromosomal DNA. To increase the number of copies of the alpha amylase gene that were present on the B. subtilis chromosome, the chromosome transformants were grown, and the presence of high levels of chloramphenicol, only cells in which spontaneous DNA duplication of the integrated plasmid had occurred could survive under these conditions. The cells selected for a high level of chloramphenicol resistance were then assayed for alpha amylase activity, as shown in 6.7. With the selection procedure, cells with up to 9 copies of the alpha amylase gene were identified. The level of enzyme activity expressed from the chromosomally integrated genes far exceeded the activity levels that occurred when the alpha amylase gene was present on a multi-copy, about 20 to 40 copies per cell. B. subtilis plasmids are probably reflecting either the instability of the multi-copy of plasmid or the energy drained on the transformed cells imposed by the synthesis of the chloramphenicol resistance gene and alpha amylase. In one study, several copies of a foreign gene were inserted into different predetermined sites on the B. subtilis chromosome. To do this, a two-step procedure was used for each copy of the foreign gene to be inserted. First, a selectable marker gene, such as antibiotic resistance gene, was inserted in the middle of a defined but non-essential piece of B. subtilis chromosomal DNA on a plasmid vector that could not replicate in B. subtilis, as shown in figure 6.26, step 1. The following transformation of B. subtilis with disconstruct cells expressing the marker gene was selected. These transformants carried the selectable marker gene integrated at the specified site of the B. subtilis chromosomal DNA. Second, the target gene with its transcriptional and translational control sequences was inserted into the middle of the piece of B. subtilis. chromosomal DNA that was used with the marker gene and introduced into the cell as a non-replicable plasmid, as shown in figure 6.26 step 2. Following the transformation of B subtilis with the target gene, plasmid constructs cells that no longer express the marker gene was selected after replica plating. These transformants carry the target gene and not the target gene integrated into the subtilis B subtilis chromosomal DNA to integrate additional copies of the target gene into the host. So the figures are the insertion of foreign gene into a unique predetermined site on the B subtilis chromosome. And step 1, a marker gene is integrated into the host cell chromosomal DNA by homologous recombination. And the former diagram shows clone gene, plasmid DNA, clone gene, its integration of a clone gene into a chromosomal site. The clone gene has been inserted on a plasmid in the middle of a clone segment of DNA. AB from the host chromosome homologous DNA pairing occurs between plasmid-borne DNA regions A and B and host chromosome DNA regions A' and B'. Respectively, a double crossover event, X to X, results in the integration of the clone gene. B. The clone gene is inserted adjacent to the clone DNA from the host chromosome. C. Homologous DNA pairing occurs between plasmid DNA region C and host chromosome DNA region C'. A single recombination event within the paired CC' DNA region results in the integration of the antiplasmid, including the clone gene. To integrate additional copies of the target gene into the host cell chromosomal DNA, the entire procedure was repeated several times with different defined but non-essential regions of the B subtilis chromosomal DNA. We're going back to the diagram. Insertion of a foreign gene into a unique predetermined site on the B subtilis chromosome. Right? The integration of a selectable marker gene along with the gene of interest is helpful for identifying transformed cells under laboratory conditions. However, the presence of a selectable marker gene for antibiotic resistance in a genetically modified organism that is released into the environment is not desirable. For organisms that are intended for deliberate release into the environment, example to degrade pollutants, it is desirable to avoid the concomitant proliferation of antibiotic resistance genes in the environment to ensure that only the clone gene is integrated into the host chromosome, the clone gene may be inserted into a DNA fragment on a plasmid that is identical to DNA in the host chromosomes. Moreover, the input DNA sequence must be targeted to a specific non-essential site within the chromosome, or important host functions will be disrupted. However, when no non-essential host genes have been identified, a clone gene may be introduced into the host chromosome by means of a single crossover that incorporates the entire input plasmid into the host chromosome. This occurs when the clone gene is inserted on a plasmid next to the cloned chromosomal integration site. In this case, any selectable markers on the plasmid, including antibiotic resistance genes, will be inserted into the host chromosomal DNA. To avoid the problems associated with these approaches, several groups of workers have developed insertion removal systems for the selective removal of marker genes from host cell chromosomes. An overview of one of these systems is described below. When a marker gene is flanked by certain short-specific DNA sequences and then inserted into either plasmid or chromosomal DNA, the gene may be excised by treatment of the construct with an enzyme that recognizes the flanking DNA sequences and removing them. One combination of an enzyme and DNA sequence that is useful for the sort of manipulation is the Cri-Loxp recombination system, which consists of the CRA recombinase enzymes and two 34 base pair LAXP recombination sites. The marker gene to be removed is flanked by LOXP sites, and after integration of the plasmid into the chromosomal DNA, the marker gene is removed by the CRA enzyme. A gene encoding the CRA enzyme is located on its own plasmid, which can be introduced into the chromosomally transformed host cell. Marker gene excision is triggered by the addition of IPTG to the growth medium. This represses the LAC1 gene, encoding the LAC repressor, which turns the E. coli LAC promoter operator, which was present upstream of the Cri gene, and causes the Cri enzyme to be synthesized. Once there is no longer any need for the CRA enzyme, the plasmid that contains the gene for this enzyme under control of the LAC promoter may be removed from the host cells merely by raising the temperature. This plasmid has a temperature-sensitive replicon that allows it to be maintained in the cell at 30 degrees centigrade but not above 37 degrees centigrade. All the protocols for the excision of marker genes have not as yet been widely implemented. Most of the details have been worked out, given the unease that exists among certain segments of the public in various locales regarding the deliberate release of genetically engineered bacteria into the environment. It is essential that the organisms that are released be as benign as possible. Removing antibiotic resistance genes is an important step in that direction. Increasing secretion. The process of secretion of proteins in E. coli entails exit through the inner cytoplasmic cell membrane to the periplasm of many proteins and passage through the outer mechanism for a few proteins. Directing a foreign protein in the periplasm or the growth medium makes its purification easier and less costly, as many fewer proteins are present there than in the cytoplasm. Moreover, the stability of a clone protein depends on its cellular location in E. coli. For example, a recombinant proinsulin is approximately 10 times more stable if it is secreted, exported into the periplasm than if it is localized in the cytoplasm. In addition, secretion of proteins to the periplasm facilitates the correct formation of disulfide bonds because the periplasm provides an oxidative environment, in contrast to the more reducing environment of the cytoplasm. Table 6.8 indicates the amounts of secreted recombinant pharmaceutical protein attainable in various bacterial systems. Table 6.8 shows the yields of several secreted recombinant proteins produced in different bacteria. The protein, yield per liter, and host bacterium. I'll just read out the names for you. Hirudin, human antibody fragment, human insulin-like growth factor, monoclonal antibody 5T4, humanized anti-CD18 FAB prime 2, human epidermal growth factor, alkaline phosphatase, staphylokinase, human proinsulin, human calcitonin precursor, organophosphohydrolase, human CD4 receptor, and human insulin. Dear friends, as you listen, please remember, my reading out every detail in this is also meant for those with impaired vision as well, but who can hear and speak well. So please do note every point, and if you can, do share these study materials with those who still have no idea that this is available here on YouTube, which is a great thing for YouTube to help us propagate the subject of study for all of you who are interested in higher professions and bettering or augmenting your career. All right, proceeding with the subject. Secretion into the periplasm. Normally an amino acid sequence called a signal peptide, also called signal sequence or leader peptide, located at the N-terminal end However, protein facilitates its export by enabling the protein to pass through the cell membrane. It is sometimes possible to engineer a protein for secretion to the periplasm by adding the DNA sequence encoding a signal peptide to the cloned gene. When the recombinant protein is secreted into the periplasm, the signal peptide is precisely removed by the cell secretion apparatus so that the N-terminal end of the target protein is identical to that of the natural protein. However, the presence of a signal peptide sequence does not necessarily guarantee a high rate of secretion. When the fusion of a target gene to a DNA fragment encoding a signal peptide is ineffective in producing a secreted protein product, alternative strategies need to be employed. One approach that was found to be successful for the secretion of the protein interleukin-2 was the fusion of the interleukin-2 gene downstream from the gene for the anti-propeptide maltose binding protein rather than just